This is the Neurosurgery Podcast.
Welcome back to the Neurosurgery Podcast. I am super excited to have with me Ashish Shah. Ashish actually is one of the Miami boys. Ashish, welcome to the podcast.
Thank you so much. I'm glad to be here.
Now, I wanted today to go into a little bit of geeking out on research and what you're doing, and I've heard you give your talk on oncogenic viruses and the theories that are at play here. And I'll just tip my hand. I studied microbiology, immunology at Stanford. I worked at CDC. I was doing an MPH in 1989. We
should get you in the lab.
Yeah, I know, right? Virology and its relationship to brain tumors, right? So
right.
First, tell us like how you got on this train.
Yeah, I've always been interested in viruses. I mean, if you look, it's a great question. I think if you look at the history of cancer and how people have studied cancer, there's always been a link to viruses. In fact, one of the first viruses, retroviruses ever found, was found to actually cause cancer in
Burkitt's lymphoma.
Burkitt's lymphoma is one of them. The other one is cause cancer in fowls, like in chickens.
Yeah.
So the first Nobel Prize given for a cancer-causing virus was in the 1950s.
Wow.
And so I was always interested in history, and it was always interesting to see the intersection between viruses and cancer formation, and then on the other side, people were using viruses to treat cancer back in the 1800s. And so I thought that intersection was very fascinating. I felt like as in dealing with brain tumors and neurosurgery, it was only a relevant thing to kind of use something that we studied from centuries ago and kind of try to revitalize it. And I thought that this was a good way of treating brain tumors.
Yeah, I mean, viruses have evolved with all living organisms, and whether they're alive or not is another question, but certainly with humans since our origins, right, as a species. So tell us a little about the theories at play, and I know that, these are not necessarily proven, but tell us a little bit about what is that relationship? Let's talk first about viruses causing brain cancer.
Yes, so this is a kind of a widely controversial topic that some people say half of us are kind of don't have the data to prove it, but I think people have been studying this for the last 50 years or so. The first studies were looking at kind of EBV or Epstein-Barr virus or CMV, these kind of herpes viruses, and looking at the association between that and glioma formation, and they found that there's viral remnants kind of in our brain tumors that we take out, and that got people really excited about this idea. That actually made a phase 3 trial studying this, in glioblastoma that ultimately failed. But if you look kind of deeper in the roots, you can actually, use some deeper sequencing to find that certain viruses are actually associated with brain tumor formation, and that's what our lab's studying right now. We're looking at something called human endogenous retroviruses, which are viruses that me and you have. 8% of our genome are made of these viruses.
8% of our genetic data.
Yeah. So back when we were old world monkeys, we basically picked up these retroviruses as we evolved, and they basically became junk DNA. And most people say, most of your body's junk DNA, and 8% of them are formed by viruses.
Wow. Now, is it only retroviruses? How about DNA viruses and RNA viruses? Can any kind of virus potentially be involved, or is it a specific subtype?
I think certainly other viruses can be involved, and there may be some infection that happens at some point in time that may reactivate these endogenous retroviruses. Our lab is specifically interested in these endogenous retroviruses, but for example, HIV, CMV infections can all reactivate these in your brain, and COVID, for example, can also do this too, which makes it very little scary to think about, how many people got COVID infections.
Yeah, it's very interesting. So let's go a little deeper, right, for the people who actually know something about medical science and all that, right? So you're studying GBM, glioblastoma, right?
Correct.
Which is not a single disease, right? It's a very complicated, probably a family of cancers, right? And what exactly is the theory behind the mechanism, the mechanistic piece of how the virus either triggers or exacerbates or creates GBM?
Yeah, so usually there's some sort of triggering event that we don't know what that is, right? Whether that be a viral infection, a flu, a CMV infection, trauma, radiation kind of incites these endogenous retroviral sequences, so these kind of remnant viral sequences in our body then become re-expressed. What these virus sequences do is they can actually insert themselves into the genome and cause mutations. The other idea is that these viruses have protein coding regions that actually can cause oncogenesis. So when these proteins are formed, they suppress the immune system, they cause your T cells not to work, they increase the stem-like cells in the GBM to actually become more resistant to chemotherapy, and they can form into different types of other cells. So you're basically promoting a kind of a stem-like niche in the tumor, and that's what we think is happening.
So I heard you mention at least three different possibilities. One is that the actual DNA that's inserted is part of the cancerous DNA, right?
Correct.
Another was that the ecosystem was altered mainly maybe through immune system dysregulation,
Right.
allowing normally suppressed cancers to grow, right?
Right.
And the third is that it would take a cancer across some kind of a asymptotic growth phase that becomes a clinically relevant entity.
Absolutely, yes. So I think that these viruses, whether their RNA can mess with their own DNA, and integrate. If you remember, kind of from back in kind of high school biology, that this is the reverse transcriptase. The RNA goes to DNA and it goes kind of inserts itself into our genome. We call that insertional mutagenesis. So that can actually go and insert itself in different parts of the genome, and that can really mess up our tumor suppressor genes and all these other factors. And then if it makes protein, that protein itself can cause the T cells or immune cells not to work, and it can also cause the tumor to grow and proliferate more. So kind of understanding that biology is important, and no one's really studied this because they think these are junk DNA. This is junk viral DNA.
They don't even look at the sequence, right? It's irrelevant sequence, right?
It's irrelevant sequence. If you look at the TCGA or you look at the Cancer Genome Atlas, no one looks at these at all because they think this is not important.
Yeah, fast. I mean, you're certainly onto something exciting, and may even lead to some other avenue of discovery beyond what we were talking about today, right? Yeah. I mean, we carry all that DNA with us. It's crazy.
People are studying this now in aging, cancer, autoimmune diseases, and now there's one of the biggest startups now is actually studying these what we call retroviral DNA and understanding how we can target them.
I don't want to talk about AI too much because it's so mundane now to discuss, but computational power.
Yeah. Maybe
you'll put it that way. Has that been integral in this kind of study? Because these sequences are how many millions of codons, right? I mean, it's a lot to go through.
So now, if you think about how rapid our ability to sequence the genome and understand the deep sequences is important, because if you do superficial sequencing, you don't pick up on any of this stuff.
Yeah, describe that difference, because I think people are like, I sent my blood out or swab to get myself sequenced, or my dog, right? Yeah.
Yeah, those are very, very superficial sequences. The cheaper it is, the more superficial the series sequencing is. So you're only looking at a certain amount of base pairs in your genome, and looking, okay, well, now I'm looking at, the actual DNA, but then if you go deeper, you're looking at the RNA. And if you want to get even deeper, you can look at all the non-coding RNA. All the RNA that we think is junk, you can actually sequence every single one of those areas. And you can sequencing to like a very, very high reading depth. So you can say, okay, well, yeah, you have a high amount of X, Y, and Z viral burden in your body that you've had that is different from someone else. And we're trying to understand, what makes certain tumors have a high viral burden versus others. And I think you have to have the right computational bandwidth. You have to have computational biologists kind of work with you. We work with NIH a lot. They have pipelines to do all this, and so.
I'm always so humbled when I talk to you, because I think about how smart neurosurgeons are in general and how much smarter you are than I am about all this stuff and how much I've forgotten. But I remember the discussion when Craig Venter was honored guest, I think at the CNS, and he was talking about the Obi-Wans, the pluralized PCR, reactors, and he was like, I'm just going to build armies of Obi-Wans, these machines, and now we're going to sequence cheaply. Is that what we're still doing? Is there a new way to figure this transcriptional information out?
So there's like chip-based assays now. So the back old qPCR, the old days of when you have a certain gene you want to look at, you amplify that and kind of sequence that. Now now we have these. The sequencing takes so much quicker to do.
Yeah.
And the Illumina is like one of the big platforms that people are using, and now we're moving to faster platforms. You can even get these reading depths within within 20 minutes to an hour.
Wow.
Even in surgery, you can take this up from surgery, get it sequenced. The question is now to read that sequence and interpret it is limiting. But I think we're going to get there very quickly. Craig Venter actually just passed away.
Oh, did he?
My
God,
week,
he's genius.
Yeah.
Yeah.
So big loss in the field. Yeah.
Yeah, he's like the Richard Branson of that world, right? He's also exciting and cool. Like Elon Musk is kind of like that. Wow, I remember him speaking. I was inspired. Of course, in spine we don't really have that kind of stuff. But so let's now take the other side of the coin. So viruses may have a hand in causing symptomatic cancer. How are you also looking at viruses as a treatment modality for brain cancer?
Yeah, and this kind of stems from the idea that you have to use biology to fight biology. And I think, if you think from a pharmacological perspective, you treat a drug, the cells in GBM resist that drug. You have temozolomide, alkylating chemotherapy, that cell will adapt to that therapy.
Okay.
Viruses have evolved to survive and adapt in bad states, and they figure out a way to survive because that's what they have to do. They're parasitic organisms,
Right.
And so we decided to use viruses because we think that we can induce an immune response, adapt with the biology of the tumor, and then you can use it, iteratively to try to treat the tumor as it evolves. And so that got me excited. Nori Kasahara was at Miami when I was a resident, and I was really fortunate to work in his lab.
Where's he now?
He's in UCSF.
Okay.
So he was in Miami, and he was developed one of the first replicating retroviruses to be used in humans. And he had a phase 3 trial we enrolled patients in Miami, and I was fortunate to develop our own retrovirus at that time. And that got me excited because they had clinical benefits in certain subset of patients. And so, of course, in my naivete, I said, okay, well, maybe I can do the same thing and try to replicate that data here in Miami. And we actually started a phase 2 trial in Miami.
Oh, wow.
Using that same retrovirus in recurrent GBM and using it in a novel way.
Yeah, so just like viruses are generally species specific, right? They could maybe be specific to the tumor part of the human body, like that.
So it's actually interesting. So this retrovirus is scary because you definitely don't want to inject retroviruses into the body because they can integrate and they can cause cancer on their own. This is a very specific one because the cells aren't replicating in the brain.
Okay.
So in order for retrovirus to get in, the cell has to divide. So because our neurons are not dividing and the nucleus is not open, these retroviruses can go into the glioma cell specifically, replicate only in the tumor cells without causing off-target effects.
Well, that's brilliant, but you got to geolocate it there.
Exactly.
And that's where the surgeon comes in.
That's where the surgeon comes in.
Okay.
You can't. We inject IV as well, but it doesn't get to the brain as much as we want to get, in direct injection.
Yeah. Okay. Well, before we close, I want to touch on a point that's I think important for being a message to our listeners writ large. You run a busy wet lab. You have translational clinical research. You have a huge following of students, and when I say students, including postdocs and PhDs and all that, and you're leading that charge. My sense is that neurosurgery is losing touch with its roots in fundamental mechanistic research, and folks like you are keeping that flag in the sand, right? Tell us about this journey. And I know it's a whole topic of discussion, but a lot of people listening, they're still young and they're naive, or they're still purists in this thinking, and you wonder what happens as people get older, right? They're too busy or whatever. Tell us about how you managed to maintain that fire of creative and curious energy.
Yeah, I think and I'll say this, I think I started probably on the other side of things. I said I wanted to do clinical research and kind of focus and building my skills as a surgeon, and then as I went through residency, and I think Alan Levy, and what an amazing chairman, he sat down with me and said, listen, what are you going to do when you become faculty? How are you going to really, be in tumors? And I said, I want to be involved with translational research. Well, what does that mean, right? And so, for me, I was like, well, if I want to really continue my viral work, I have to really dig deeper and understand the mechanisms and biology of what I'm doing so that I can then translate that to patients. So that became a quest. The more I read, the more I kind of wanted to answer these questions. And so I went to NIH for a year after residency, and that, that was a move because I didn't have the translational basic science background I needed, right?
Right.
And I went there for a year, picked up an amazing skill set, made amazing collaborators, and these people continue to ask you questions that you're like, well, I don't know that answer. I mean, how can I be doing this research if I don't know that answer? So then we answer that question in the lab, and then the question begins another question. And then as you discover more and more, and you see actually cures in mice, you're actually curing brain tumors in mice, you said, okay, well, listen, we can actually bring this to patients and bring it to them easily, right? So that's what kind of got me interested, and it kept on going. And I told myself I'd never run a Western blot after medical school. I said, I'm never running this again. I hate Western blots. Yeah. What a horrible situation. And now all my lab does is run Western blots. And I realized that, it's kind of a necessary evil.
Well, Dr. Shah, listen, you have inspired so many young people to work with you. Not only are you a great researcher, a great clinician scientist, and a great surgeon, but also a great communicator. So folks listening that are interested, they want to reach out to you, they want to support your laboratory efforts. How do they reach you at the Shah Lab?
We have a website. We're on Twitter. We're on Instagram. So
give us the handle.
It's just SVI Lab.
SVI.
Yeah, the section of virology and immunotherapy. Yeah,
okay.
So SVI Lab at or theshawlaboratory.com. Yeah.
The Shah Lab. Wow. Excellent.
All right. No, thank you for having me. It's been a pleasure. I've been following the podcast for a while, so I'm honored to be on it.
Thanks for coming on, Ashish.
Thanks a lot.